Aloha everyone, my name is Clyde Tomorrow. I'm an aquaculture specialist with the College of Tropical Agriculture and Human Resources, or CITAR. I'm also the principal investigator of this CTSA supported project that's entitled Alternative Methods for Marine Copepod Production in Hawaii. I would like to take this opportunity to recognize the other project workgroup members, and they are Karen Britton, Benjamin Alexander, or Bo Alexander, all of whom are part of the Hawaii Institute of Marine Biology, Mr. James Jackson, who is a graduate student in the Department of Zoology, and Dr. Petra Lentz, who is with the Pacific Biomedical Research Center. There's a bit of history that comes with this project, and originally the PI was Dr. Jim Shipper, who was at the time with the Sea Grant College program, and he was working out of the UH Halo's Pacific Aquaculture and Coastal Resource Center. However, budgetary constraints resulted in an earlier than anticipated retirement, leaving the project unfinished. A call went out from CTSA for interested parties that would be willing to complete the project, and our proposed work plan was selected and in correspondence dated August 30th, 2010 from Dr. Cheng Seng Lee. CTSA would approve our revised work plan, which forms the basis for the current project and report. Because of the delay in completing the project, there were a couple of stipulations, and they were: the project period was to begin January 1st, 2011, and would end on December 31st of 2011. The funding level was restricted to $25,106. As with all of our projects, we form collaborative partnerships where the intellectual capital and the resources of various participating institutions all can be brought to bear on achieving the desired outcomes. So in this case, with funding from CTSA, the participating institutions were CITAR, Hawaii Institute of Marine Biology, the Department of Zoology, and PBRC. So why have this project? The goal of the project was to make marine fish production in Hawaii less costly and more available for consideration by businesses. Uh, this was to be done by developing and communicating new or newly adapted culture technologies for marine copepods. This is an important class of live food item for rearing the early larval stages of marine fishes, both food fish and also marine ornamentals. Now, copepods are excellent live feeds for most larval marine fishes and are often associated with significant advantages in survival, early growth rates, and also color development over the more commonly used rotifers and brine shrimp artemia. The use of copepods, and particularly their early life stages, the nauplii stage, as the initial larval feeds, has generally improved survival over other rearing regimens, and there are also some fishes who require copepod nauplii as a live food source to ensure success in early larval rearing. Successful adaptation of the culture practices would permit new and smaller businesses to examine copepod culture, and therefore production of new species at reasonable costs and low levels of investment. So copepods in general, and particularly the nauplii stage, are generally more costly to produce per unit than rotifers are. This is mainly related to the copepod's lower tolerance for high density culture. Both are typically fed cultured live microalgae, which requires both skilled technical staff and, as you can see with the photographs on the right, a considerable amount of infrastructure. However, considerable progress has been made on the use of commercial algal paste, such as instant algae, a product of reed mariculture, that can actually replace the live microalgae requirements for a host of hatchery operations, such as rotifer production. Adapting the use of the algal paste for the culture of copepods became an obvious area that would need investigating. An example of a place-based impact of having alternatives to live phytoplankton cultures are the recently concluded hatchery outputs of the Pacific threadfin moi, or Polydactylus sexphilus, that were done under the auspices of a collaborative project with Hukilau Foods at the Hawaii Institute of Marine Biology. In short, the trials resulted in a consistent harvest density of over 20 fry per liter at 25 days post-hatching, and this is a remarkable production of 60,000 fry from a single 3,000 liter rearing tank. Not only was this achieved on three consecutive trials, but more remarkable, it was done completely without the use of any live phytoplankton culture. Alternatively, work also done at the HIMB with developing technologies for the artificial propagation of the feather duster worm, an important marine ornamental, has revealed that one of these commercial products has also utility for non-traditional species like the feather duster worm. In this case, the feather duster worm, we used a commercial paste for Isochrysis galbana, and it was found to be a suitable substitute in terms of resulting survival and growth of juvenile feather dusters when compared to live Isochrysis, as well as other live phytoplankton species. The successful adaptation of the use of this product obviously provides for an opportunity for expanding and diversifying the aquaculture industry in the form of culturing this marine ornamental. So the overarching question that needed to be answered is, can calanoid and herpactocoid copepods be adapted to low-cost culture methods using artificial seawater and also artificial feed particles? And to answer this overarching question, several objectives were to be addressed over the course of the project. The first was to assess the adaptation of copepod cultures to artificial seawater and also artificial feed particles. This would then be followed by demonstrating that the culture technology could be expanded and to achieve an intensification of copepod cultures that would result in nauplii and adult densities that were equal to or more than 20 animals per milliliter. Likewise, the culture volume and therefore production outputs had to also be expanded beyond the laboratory scale 1 to 2 liter size range up to the larger 300 to 500 liter volumes, using artificial seawater and also artificial feeds. The remaining challenge would be to demonstrate that the techniques have utility over the course of a long term, measured in months rather than just days. And lastly, as with all CTSA supported projects and also part of CITAR's mandate as a land-grant college, we would need to make sure that the technologies are disseminated to appropriate end users. The classical means of transferring technology comes in the form of technical handouts, newsletter articles, and of course the ever popular workshops are to be employed. The last live feeds workshop that was held at HIMB is shown in the photograph on this slide, and that was completed on March 20th of 2008. So the first question that needed to be addressed was whether or not the source of seawater, natural or artificial, would have an impact on phytoplankton production. Although most mariculturists use natural seawater, the use of artificial seawater continues to play an important role in the culture processes because of the advantages of having a known chemical composition that can be consistently produced. Natural seawater is not readily available to inland areas, and while there are a host of different recipes for making artificial seawater, we chose to use only one, and that's Instant Ocean, because it's well known and a readily available commercial product. The use of this commercial product is not an endorsement, and the drawback to its use and for other recipes in producing artificial seawater are its obvious costs. Trials were conducted in 2-liter bottles using standard protocols that have been established at the Hawaii Institute of Marine Biology Bottomfish Hatchery Facility with regard to the micronutrients that are added, aeration rates, sterilization procedures. The only difference in the culture process was a source of seawater which was used, and one being artificial seawater and the other natural seawater filtered and processed from Kaneohe Bay. 3 species of phytoplankton, Isochrysis galbana, Chitoceras, and Tetraselmis, were tested in replicated trials, and the temporal changes in cell density were monitored using a compound microscope and hemocytometer. Results of a trial where Isochrysis galbana was used as the test microalgae is summarized in the graph on the right hand side of this slide. Such results were typical of all of the species investigated, and in summary, there were no differences in the use of artificial and natural seawater in production of the various phytoplankton species that were investigated. 2 species of copepods were the target of the investigations during the project period. The first was a calanoid copepod, the species Parvocalanus castorostris, and the second is the herpetocoid copepod, Euterpina acutifrons. Investigations testing the various treatments were conducted in 5-gallon plastic buckets that were done in a static fashion but were supplied with a continuous source of aeration. When a treatment called for the use of artificial seawater, the phytoplankton that was used as the food source had also been cultured in artificial seawater as well, so everything being kept constant. Temporal changes in copepod densities were quantified by using a glass pipette and taking 1 mil ethoquats, a minimum of 4 per sample period, and then using an 8x lube, the various stages of copepods present within the pipette could be counted and recorded. Numbers of adults, copepods, and copepodites have to be grouped together because they're very difficult to distinguish from each other, but that's not the case with the easily differentiated Naplea stage. Algal cell densities were kept at 200,000 cells per mil and were provided throughout the course of the experiment. The initial experiment that examined the use of artificial versus natural seawater for culturing calanoid copepods took place over a duration of 14 days. The temporal changes in Naplei that you can see on the top graph on the right and the adult copepod densities, the bottom graph also on the right, that took place during the trial is summarized. The data was subjected to statistical analyses, and the observed mean Naplei densities for those that were cultured in natural seawater were found to be actually significantly higher than when using artificial seawater. It should also be pointed out that the highest average Naplei densities obtained when using artificial seawater was still over 20 individuals per mil, which is actually a suitable quantity to obtain when conducting larval rearing trials. The exact reason for the higher production of Naplei in the natural seawater is not known. In contrast, when we took a look at the mean number of adults that are observed between the two different sources of seawater, they were found not to differ statistically, as the average total number of individuals was also found not to differ significantly. So in conclusion, although there's subtle differences in Naplei production, overall it would appear that there's really no difference in using artificial or natural seawater for culturing this particular calanoid copepod. When the herpetocoid copepod was also tested, it too was found to perform similarly, and there are no differences that could be detected when the culture was being done in artificial or natural seawater. A separate series of experiments using the herpetocoid copepod was undertaken, where this copepod was provided with a variety of phytoplankton species. This was done in order to assess their growth performance using all artificial seawater conditions. Results of the trials are summarized in the graph on the right, and on the top graph you can see Naplei densities of 20 individuals per mil were recorded using both Tetrasomus and Chitostris as a food source. But no differences in performance could be statistically detected. A similar situation was also observed with the adult populations. However, when we take a look at Naplei production, it was significantly less when we used Isochrysis galbana as the food source, and this is also translating to a much significantly lower output of adults. The use of artificial seawater can obviously support the growth of this herpetocoid copepod, and it appears that the choice of the phytoplankton species that is used as the source of food is going to play a more significant role in their culture than the actual source of seawater. A similar kind of trial, as shown in the previous slide, was also conducted with the calanoid copepod, but in this case only two species of phytoplankton, Isochrysis galbana and the Chitostris, was provided at 200,000 cells per mil. Temporal changes in Naplei and adult densities are summarized in the graphs on the right hand side of the slide. The results obtained were consistent with the previous results when using natural seawater, in that Chitostris does result in growth of the calanoid copepod It does not perform as well as the production of Naplai that later translates to overall production of adults in comparison to when Isochrysis galbana is used as the food source. As with the herpactocoid copepod, the choice of phytoplankton apparently plays a more significant role in overall production outputs for this calanoid copepod than does the source of seawater. Because of the inherent costs of using artificial seawater, one strategy that we took into account is that we might be able to dilute the solution in order to maximize its use. Thus, knowing the effects of varying salinities on calanoid copepod production was going to be very, very important to know. So culturing the calanoid at various salinities was conducted by diluting the artificial seawater using tap water that resulted in the five different salinity treatments that you see on the right. All treatments were conducted in triplicate and as static systems that were provided with a continuous source of aeration. Isochrysis galbana, also cultured in artificial seawater, was used as a source of food and provided at a density of 200,000 cells per mil. The results obtained over the course of a two-week grow-out period is summarized in the graphs on the right-hand side of the slide. Both Naplii and also adult calanoid copepods did not grow well below salinities below 20 parts per thousand. Clearly, both Naplii and adult production was highest when using the highest salinity or the 30 parts per thousand treatment, and indicates that the range of salinities that would be optimal for copepod production is going to be restricted to the higher salinities or 30 parts per thousand and above. Commercial algal paste such as Nanochloropsis oculata that's produced by reed mariculture has been shown to be highly effective in supporting rotifer production to the point where live phytoplankton can be totally replaced with the use of this product. Likewise, the company produces a variety of other algal pastes that have been reported to be suitable for culturing other species of organisms. We gave the example of feather duster worms earlier. The results of trials using a calanoid copepod grown using a variety of these algal pastes is summarized in the graphs on the right hand side. Unfortunately, all of the commercial algal pastes that were tested failed to result in any significant ability to support the calanoid copepod growth. So this is a similar experiment as that reported in the previous slide, with the big exception being that the herpactocoid copepod was the target species in this trial. Results are the same that we obtained with the calanoid copepod, where we couldn't find any kind of supportive growth using any of the algal pastes that were used. So we also tried some alternative feeds that were investigated as to their abilities to support growth and production of both calanoid and herpactocoid copepods. Attempts at using some of the more innovative artificial feeds like V8 vegetable juice that's been reported to be successful for another marine herpactocoid copepod, as well as crushed nori was also attempted. Results are summarized in the graphs that are on the right hand side of the slide. And in short, both the V8 vegetable juice and crushed nori did not result in any production of the target copepod species being investigated. This is contrary to some of the results that have been previously reported. To investigate the long-term performance of copepods that are going to be grown in artificial seawater conditions, we used a 40-liter culture vessel using only artificial seawater for both culture of the phytoplankton and the calanoid copepod. The experiment was initiated on December 2nd in 2010. The culture was run as a static system and the salinity ranged between 30 and 33 parts per thousand. During this time period, the copepods were supplied with a continuous source of aeration, and also Isochrysis galbana was used as a source of food. Phytoplankton was provided at a density of 200,000 cells per mil on a daily basis and produced all using artificial seawater. The density of Naplii and adults were monitored as previously described, and at approximately 2 weeks duration, the culture is completely drained, tank cleaned, and the culture restocked using the same individuals that were harvested. A summary of the temporal changes in Naplii and adult density is provided in the graphs on the right hand side of this slide. The typical bloom of nauplii that occurs with each restocking of the tank is routinely observed, followed by a rise in the number of adults. This nauplii bloom is one of the mechanisms that can be used to provide the nauplii stages of the copepod to first feeding fish larvae that require this particular live food organism stage. Clearly, the culture of this calanoid copepod can be done using only artificial seawater and also over extended periods of time. Future work should include focusing on the recycling of the artificial seawater, as it represents a substantial investment and warrants its reuse. So two deliverables are going to be used to satisfy the technology transfer objective, and one is a newsletter article that has already been printed in the June 2012 issue of CTSA's Regional Notes. The second deliverable is this narrated presentation that's going to be placed on CTSA's website, but also will be placed on my website that's housed in CITAR. A significant delay in obtaining the commercial algal paste due to permit acquisitions, although has been resolved, really delayed the completion of the last technical task that needed to be completed. A technical handout is being planned, but it is dependent on obtaining additional funding for the work that's to be conducted at HIMB. Likewise, a workshop is also being planned after the sequestering of additional funding. So summarizing then, there were no detectable differences in the growth of 3 microalgae when grown in natural or artificial seawater. Likewise, when culturing both the herpetocoid and calanoid copepods, there are no apparent differences in the culture performance with the use of natural or artificial seawater. When you combine the results, this clearly indicates that the availability of natural seawater is not a limiting factor for copepod culture. The culture of the calanoid copepod indicates that the optimal salinity for its culture is 33 parts per thousand or higher. The focus is then now on the next steps should actually figure out how to recycle the artificial seawater after it's used because of tremendous amounts that are being used. Clearly, the kinds of phytoplankton that's used in the culture of the copepods has a much more profound influence on culture performance than does the actual source of the seawater. So results that have been obtained to date demonstrate that not only can artificial seawater be used as a culture medium for producing copepods, but the culture process can actually be conducted over an extended period of time without any adverse effects. What was disappointing, however, was not being able to find an alternative feed particle that can replace live phytoplankton as a source of food for the copepods. Obviously, this area is an area for future research. In closing then, I would like to extend my sincere mahalo to the project workgroup members for making this presentation possible, and likewise, mahalo, thank you for taking the time out to review this presentation and the results of our work. If there are any questions, do not hesitate to contact me, and my contact information is located on the bottom left hand corner. Aloha!